Flow cytometry is an immunoassay technique used to phenotype single cells in suspension. This video will teach you how to stain your cells with fluorescently labeled antibodies and viability dye for flow cytometry analysis. Cells need to be in a single cell suspension at approximately 1 to 10 million cells per milliliter. Dead cells increase background through autofluorescence and nonspecific antibody binding. Include a viability dye to measure cell viability and exclude dead cells from downstream analysis. Add 1 microliter of phantom dye solution for each milliliter of cell suspension and vortex the cells immediately. Incubate cells with phantom dye for 30 minutes at 4 degrees Celsius in the dark to prevent photobleaching. Cells should have over 90% viability and not less than 95%. To remove unreacted dye, wash the cells once with 1 to 2 milliliters of stain buffer made up of PBS with 2% FBS and 0.09% sodium azide. Pellet the cells by centrifugation at 300 G for 5 minutes. Nonspecific binding of antibodies to Fc receptors in your sample can cause high background signal in your data. To reduce background, resuspend your cells in blocking buffer containing 10% serum. You can skip this blocking step and produce clean flow cytometry data by using ProteinTech's Fc0 recombinant antibodies containing a mutated Fc receptor binding region. For all other antibodies, incubate the cells with blocking buffer for 5 to 10 minutes at 4 degrees Celsius. Centrifuge the cells as before, discard the supernatant, and resuspend in PBS for antibody staining. For antibody staining, add 1 million cells per condition to fax tubes or to a 96-well plate with a U-shaped bottom. In addition to your samples and biological controls, prepare compensation controls and an unstained sample to assess autofluorescence. Consider using isotype controls to compensate for nonspecific binding, and fluorescence -1 controls to establish accurate gating. Pellet the cells by centrifugation at 300 G for 5 minutes and discard supernatant. Antibody working concentration should be determined via titration to maximize signal-to-noise ratio. Make a master mix of your antibody cocktail to ensure all samples are stained with the same amount of antibody. Resuspend the cells in their respective antibody mixes. Incubate the plate in the dark at 4 degrees Celsius for 30 minutes to allow antibody binding. Following incubation, wash the cells twice with PBS by centrifugation at 300 G for 5 minutes to remove unbound antibody and reduce background signal. If you're using an unlabeled primary antibody, repeat the staining and wash steps with an appropriate secondary antibody. Or simplify your protocol by directly conjugating any primary antibody to a fluorophore using flexible antibody labeling kits. After the final wash, cells should be resuspended in 300 microliters of PBS for analysis at the flow cytometer. For best results, analyze your samples immediately after staining. For extended storage, fix the cells with 4% paraformaldehyde, wash with PBS, and store in the fridge for up to 1 week. Acquire enough events to ensure statistically significant analysis. For rare populations, aim to collect at least 2,000 positive events from your population of interest. For more details, check out our protocols and resources linked in the video description.